Here we go. So overall, we're doing gene regulation. That's our bottom line, right? We're studying promoters to determine what factors help turn them on and off, mostly turning on, okay? And the system that we started with to sort of decide what kind of genes we want to work on was from a study at UCLA that did a subtraction between two different types of lymphomas. One is a mantle cell lymphoma and one is small lymphocytic lymphoma. Mantle cell we chose because it's very aggressive and because we had funding from a private foundation interested in mantle cell lymphoma research. It's not a super common lymphoma, wasn't really on the radar for lots of big grant agencies, and so this small foundation really wanted to do some more work. And small lymphocytic lymphoma, which is also known as chronic lymphocytic leukemia. So if it's found as a lymphoma, which is a solid tissue mass when they find the blood cancer, it's called a lymphoma. If it's found circulating through the bloodstream, it's considered leukemia. And they claim both of these are essentially manifestations of the same type of cell. Again, who really knows if that's true? But either way, they consider this an indolent disease, which means slowly progressing or slow growing or not aggressive. Whereas mantle cell lymphoma is very aggressive. And at the time, 10 years ago, there weren't very many treatments that were at all useful for lots of lymphomas, specifically mantle cell lymphoma. And so we wanted to look at essentially gene profiles. What are the similarities between the two lymphomas? What are the differences? And we came up with sort of candidate genes that were expressed in mantle cell and not in SLL and not in mantle cell and yes in SLL. And so what we didn't do was look at what mood lighting. What are these genes, how are these genes expressed in a normal B cell of the same developmental stage? And so that's something that each one of us should look up and do a PubMed search for their gene. What is it supposed to be like? What is it in a regular B cell, not cancerous? Is your gene expressed or not? Because that tells you a little bit more about if it's on here and off here, that could be normal, right? It could be, the on could be the normal, right? If it's on in B cells, then it should be on. And the interesting thing is that it's off in SLL, or vice versa. So it really does matter its expression in a normal B cell so that we can talk about why it's different in either one of these lymphomas. So just because it was found in mantle cell doesn't mean that it's not necessarily overexpressed or aberrantly expressed or dysregulated there. It depends on what it looks like in the normal state. So that's really important for each one of us to look at our gene. And there should be some information out there, either through another type of microarray or just people doing RT-PCR or staining. If there's no information out there, then there's no information out there in the database. There's nothing we can do about it. We would just keep looking, to see what it's supposed to be like to give us a better idea of what's going on here. So these are really just differences between these two, not overexpression. Whereas, because this was done by not a normal type microarray, SSH, not SHH, suppression subtractive hybridization, it avoids the step of amplification that all microarrays use by the PCR amplification process. In this protocol, you essentially stop the amplification step. So you suppress it so that every gene, regardless of if there's tons of copies or just a few copies, will still be represented in your profile at the end. So the beauty is that you get genes that are expressed at low levels, not just genes that are overexpressed. Whereas most microarrays only get genes that are overexpressed. So you lose anything that's, is on, but just at a low level. And then sometimes in the microarrays, the amplification step, right, whichever genes are PCR amplified better tend to show up, even though those are not necessarily the only genes that are in there. So ones that didn't happen to PCR well won't show up in a microarray, whereas they'll show up in an SSH at any level because it's not required to have that amplification step. So all we can say is it's in one and not the other, or vice versa, right? But we can't say overexpressed, underexpressed, those kind of terms. So we need to watch in our abstracts and our intros the kind of wording that we're using for that, right? And then we should definitely look up what it looks like in a normal B cell of its same developmental level. Just to be pretty clear on, right, is this on aberrant or is it the off aberrant? Is the on aberrant? Is this off aberrant? Okay. So what do we care about? We were looking at genes that nobody else cares about, or there's not a lot of data out there on. Okay, we're happy if they're studying the protein and what it does, but we don't want to be competing with anybody else studying the gene regulation of, studying the promoters, right? Because there are lots of labs out there that have lots of money and postdocs and PhD students that spend 80 hours a week in the lab, and we just are not going to compete with them. So that would just be silly to choose things that other people are working on, right? So really any of the genes in our list we could look at, and the first thing we tell you guys to do is to look up in PubMed to make sure nobody else is studying the promoter. The other thing, I think in the real list in the paper, it gives you its alias names, because in searching PubMed, you generally need to search the literature for all of its aliases because they don't cross-reference each other. So if something was published 10 years ago under a name that you didn't check, that will not come up if you just search the one name for your gene. So if it's already been published, it's not something we want to do. We can't publish something that's already been published, right? So we want to choose something that's not published. So the first thing we do is we say, well, go find your gene in the database and pull the promoter region. And so if we're looking at chromosomes, right, if we have our gene of interest, right, if this is where the +1 of transcription, right, that's the first 5' in, 30' in of the RNA, and then we have to do RNA processing, which removes introns, adds a poly A tail, puts the 5' cap on it, and then, we do translation. If we're talking about protein coding genes, right? And then this is our little protein guide. So what we care about is the region of DNA just upstream of the start of transcription, right, which is this guy, right here, that first base pair that makes RNA, and this is called the promoter. And generally speaking, set up as the core promoter and the proximal promoter. Core can also be termed the basal. How do you spell basal? Basal? Mm-hmm. I was like, I-L? Basal. Right, basal promoter. So this is essentially where, right, RNA polymerase and all of his friends sit down to actually do complementary base pairing on the template strand to create the primary transcript RNA. That's not enough to do physiologically relevant levels of transcription to make physiologically relevant levels of protein. We need the proximal promoter, which, so if we're thinking about our core promoter guy, we'll do polymerase. We'll draw him there, and then he'll have a bunch of friends on him. Kind of like zits. Right? And then in the proximal promoter, there's other transcription factors that are in charge of binding to specific sequences, and those guys interact with RNA polymerase II to essentially have RNA polymerase sit down and go, sit down and go, sit down and go, and get lots and lots of transcription off, okay, of that transcript. So we're not really studying the core. I mean, the things we want to know about the core is it one of the classic promoter types in the database? Does it have a TATA box? Is it initiator driven? Does it have the DPE? Does it have all those things that are in the reviews for core promoters? And those can come out by doing that transcription factor search, and they have specific consensus binding sites, and so then we can guess what type of promoter we think it is. If there's a TATA box within range of where it should be, it's going to be a TATA containing promoter. It's going to be driven by that. That's pretty clear. The rest of them are not so much. But we really are interested more in what's going on to help the RNA polymerase go. What are all the proximal sites that drive transcription? So then what we do by the computer, right, we look for the mRNA sequence, or we just type in the name of our gene and find that. And hopefully, if we type in the name of our gene and put the term promoter, we don't get anything out of the database. Because if we do, that means somebody else has sequenced it and probably studied it, and then you have to change genes. So if you search for your gene name and the word promoter in GenBank, and it comes up with a hit and it gives you a sequence, that is likely to mean that somebody else is studying it, and that would probably be a bad idea, right? Then you'd really need to look at the literature search to see what's out there. Okay. It could be annotated based on some sort of screen. There's lots of target screens now looking at, well, what does a specific transcription factor bind? And they do a whole genome search, and they list all these genes, and they dump the sequences. So it could be totally fine, but it would be definitely something that you'd want to check on. So we use the mRNA to find the promoter sequence, and we copy and paste about 1,000 base pairs or so. What the heck is that? Or SOR. Oh my God, my eraser doesn't work. It's supposed to be an O. I don't know what the heck that was. So right upstream of the first ATG, which is what? The start of translation. Okay. We want to have, so that essentially, right upstream of the start of translation is our 5' UTR. Part of an RNA that does not code for protein. So the first ATG codes for the first methionine, that's what's translated, but that's never the first few base pairs of an RNA. There's always a 5' untranslated region. And this was essentially the very last base pair of that is the +1 of transcription, where the first base pair was complementary base paired to the template from RNA polymerase, which means anything in this direction should be that promoter if we are looking at our little, schematic from the slide before this. And so we want to take from here, right, and have this whole piece be, at least 1,000 base pairs. So we want to have as much of the 5' UTR as we can. If our primer sat right there, the first base pair beyond the ATG, great. Right? We got all the 5' UTR, and if that's 100 base pairs, then 900 base pairs, upstream of that or so. Okay. The only exception to that is if you have an intron within your 5' UTR, right, then you don't want to just start right at the ATG and take that entire piece, because we don't want, that's too big. If that intron is several thousand base pairs, then that piece is just going to be too big to deal with in our in vitro system in our plasmids. And so then we'd essentially want to take wherever that intron ends, the rest of that 5' UTR and 900 base pairs upstream of that. To make, because this, right, if this is the +1 of transcription, RNA polymerase sits here, not here in this intron. Okay, so that's one thing you need to check, especially if you were handed primers from somebody else that may not have checked or may have done it wrong, and we're actually PCRing into the intron. Okay, it's really easy to look in that database and not realize you get to the end of the purple, woo, pull 1,000, and you're actually in an intron. So it's always best for anything, any plasmids, any primers, any maps, anything anybody else hands you, check for yourself. Okay, because otherwise it's just months of work that could be based on a mistake, right? An easy mistake that's simple to make. It's not hard to make these kind of mistakes. So just check it out. And the reason we sometimes have trouble with PCR is because we want to make our primers in specific places, and we want it to be as close to this ATG as possible. We want as much of the 5' UTR. We're assuming there's no intron, right? And then this one can really sit anywhere. That one's we can, as long as it's about 1,000 base pairs, right? If you have to move up 200 base pairs to get a better primer to match with this one, great, go ahead and do that. Or what if upstream past 1,000 is very AT rich and you can't, then you make it smaller, right? You sit it at 800. But it's this one, the 3' primer, that really needs to be as close to that ATG as possible. And so a lot of times the computer programs don't like to do that, or in order to get them to find you one close there, you have to loosen the parameters so much that the PCR is going to be difficult to get to work. Okay? So historically, because of that, sometimes this primer down here is not ideal, that we chose to do the cloning the way we do by using that intermediate vector. And the most popular intermediate vector now is PGC blue, because it has the blue-white screen. Right? It's very easy. So the beauty of these intermediate vectors is they're made by a company, and they're engineered to essentially suck up a PCR product very easily. They do close on themselves, but who cares, because it's a blue-white screen. So if we have our intermediate, I'm just going to make shit up because I don't know what's really in it. PGC blue, right? And it's cut open, and it's either G overhangs on the vector and C overhangs on your PCR product, or vice versa. I can't remember. Is it season? Oh, damn it. Picked the wrong one. All right, let's change. Sorry. C's on the vector. And here's your PCR product. G's overhanging your PCR product. Okay. So the ends of your PCR product are not blunt. They don't perfectly, like that's TAC. For any of the TAC polymerases, they overhang either a G or an A. Okay, and so half of them have G's overhanging and half have A's overhanging. So the ones that have A's overhanging don't do anything for us. But the ones that have the G's overhanging, now we have this little tiny little sticky end that G's and C's will complementary base pair, right? Hydrogen bond, and then, right, the ligase we put in there will make that phosphodiester bond and hold those guys together. So if this is PCR product, which means you don't need a ton of PCR product because this almost always works. Yay! Okay. The ligation. So the ligation almost always works. Our problems come during transformation, which is the process of the bacteria taking up that plasmid, right, making it its own. This is making can? Does PGC blue
yes,
can? Okay. Right, it has the kanamycin resistance gene on it. Make it a little R, which means when it's doing transcription and translation, it's producing the kanamycin resistance proteins that when you plate it on a plate with the antibiotic kanamycin, anybody who has this plasmid can survive. Essentially, I think it's a channel protein or something that pumps the antibiotic out of the cell. I don't know. I don't remember. whatever. It doesn't like kanamycin, kill it for whatever reason. And any of the bacteria in that transformation that did not take up plasmid will be killed by the kanamycin, right? So if you think about the transformation in the little tube, that's a gazillion billion bacteria. And if you get 50 to 100 colonies, that's really good. But that means we had to kill off over 1 billion bacteria that did not have your plasmid, right? So if we didn't have that kind of selection, you'd never find, you couldn't just pick a bunch of colonies and do a DNA prep and try to find your plasmid, right? You'd have to screen 1 billion. That's a lot of boil preps. So that's the first screen to make sure it has the plasmid, okay? So any of them that close on themselves, that just, go back with their C's and, they're sad they don't have their insert, those are going to look blue, right? And that's because the beta-galactosidase LAC-Z gene, right, is cut in half here. If it comes back together, right, it makes LAC-Z, which cleaves the substrate we put on the plates, and it turns blue. So blue colonies, we know there's nothing in. It's just a vector reclosed on itself. So we don't pick those. We don't like it. We don't want it. If we get, I'm just drawing single-stranded here. Don't get crazy on me. If we get a guy that has an insert, the LAC-Z gene now, it's like having a big chunk in between, right? It's an intervening sequence. When transcription and translation starts to happen, it'll make the first few parts of the LAC-Z gene, beta-galactosidase, which isn't enough to cleave the substrate, because then it eventually will hit a stop codon. The protein is truncated, so it will not cleave the substrate, and it will not turn anything blue. So these guys stay white. So the white colonies on the plate are the ones we want to pick, because they should have insert in them. That doesn't mean it's the insert we want, but at least it's some insert, right? When we think about when we do PCR, we have specific primers, and even if you see one single band on a gel, right, we only see stuff on a gel that's in a high enough concentration to take up enough ethidium to glow over the UV light, right? If we really were looking at every single piece of DNA, if we could visualize it, every lane on our gel would be a big smear. There's molecules of misprimed and single strand and the big piece and the small piece, and where the PCR primer sat down once in a while on those, right? So if we just did a PCR cleanup, where we just take that whole tube of everything we made and clean it up and then do our ligation, it's likely some of the guys in here are not exactly what we want. But then we just screen them when we pick the colonies, grow them up, do a restriction digest from something outside. There's R1, R5, Bagel 2, or I don't know, so, a bunch of things, or do a double cut, right? We expect, if we PCR'd something 1,000 base pairs, we expect 1,000 base pair band. If there's one that's 1,200, 1,500, 500, don't pick that one, right? That's probably where the primer sat on something else. Because the beauty of cloning is you need one molecule, right? One molecule, one double-stranded molecule goes into one molecule of double-stranded plasmid, gives you one colony. So it doesn't matter if there's, 99.9% of the time your PCR products are what you want. That 0.01% that prime somewhere else could still end up on your plate because it's one molecule, right? But if you pick 10 colonies, maybe one will be wrong and the 9 will be correct. Okay, so we just don't want to assume that just because it's white, it's actually what we want. We just know there's something in there. Does that make sense? All right. I like it. Okay. So we pick all the, well, not all the white guys. Hopefully we have lots of them. And right, this has your PCR product in it. And this is PGC, I guess we call it. Yeah, that vector. So that's our intermediate vector. And at this point, we'd like to know what direction our PCR product is in, just for cloning purposes. And what do I mean by that? When we did our PCR, we want to think about if this, well, blow this guy up, right? It's our double strand. Which end was where our 5' primer sat, and which end was where our 3' primer sat? Right, our 3' primer sat, right, if the ATG in the real world, back in the chromosome, would have been right there, right, and our +1 of transcription after our 5' UTR is on that end. And so that essentially means, if we're talking directionally, this would be the forward direction of our promoter, right? This way, right, transcription's going this way. Whatever sits in front of this promoter is going to be transcribed and then translated, okay? So we can do cuts to look at orientation instead of just sequencing. Sequencing is expensive and it's kind of a pain in the ass. And we can figure out if our product is in the forward or if it's actually sitting, which we'd call reverse, right? If we're actually pointing that way, we would call this one the reverse direction. By looking at restriction sites within the sequence and outside. So if we have a bunch of restriction sites in the multi-cloning site, right, we cloned into the GC spot, and then I think PGC has Echo R1 sites right around it, and then a bunch of other crap. I'm going to make some shit up. How about that? Here's a Hindi 3 site. I know there's an XHO on the other side. I don't know which side it's on, but it doesn't really matter. And so if we're lucky, or unlucky, we have a restriction site in our sequence somewhere that matches one of the ones in the multi-cloning site. So let's pretend, let's not use one of those just because that's, we might want to clone with that. Let's do KPN because we hate KPN. KPN1, we'll pretend like it's in the vector. Right, we totally hate it. And then we're going to pretend like there's a KPN1 site. It just happens to be in your sequence, right? When you have your sequence, you can put it into whatever website you want to, or the McVector program on our computer, and ask the computer what restriction sites are in this sequence. That I directly PCR, not your original file that you pulled, exactly what's between your 2 primers. Because that's what went in this vector, right? If you pulled 2,000 base pairs of your promoter, but your primer only covered 1,000, you don't want to put in that 2,000. It'll be confusing. You want to just put in where your primers were so you know exactly what restriction sites are in there. And so if it's in the forward direction, it's sitting, right, it sits here close to the +1, which means it would sit over here if it were in the other direction, close to that +1. So if you pick 10 colonies and you prep them and you cut with R1 and they all give you the 1,000 base pairs, cut with Eco R1, you go, yay! That means we're pretty sure it's what we wanted. We expected 1,000 base pairs. We got 1,000 base pairs. Woohoo. That doesn't tell us which direction it's in. If we now cut with KPN, if we cut and it's in the reverse, in this, if we think about the vector over here would be, oh my god, go away, would be KPN on this side, and the vector still is KPN over here on this side, right? Which is this guy right here. So if we go right and it's in this direction, or if we went and it looks like that. Okay, so if any of the colonies had this set up, we're in this reverse direction, we'd get a, I don't know, I'm going to make it up, a 300 base pair piece, right? It's pretty close together right here. So we're going to say, we're going to get another color. We're going to say from here to here is 300, and from here to here is 800. So when we cut with KPN, the ones that give us a 300 base pair piece are in reverse, and the ones that give us an 800 base pair piece are in the forward direction. So we don't have to sequence to know which way it goes. And the best thing to do is to draw a map using the real background of PGC with your insert marked with relevant restriction enzymes and draw it out just like this. So like if it's reverse, here's a map. And ooh, it'll be 800 base pairs. If it's in forward, here's a map. Ooh, it'll be 1200 base pairs if I cut with this. And then you look at your gel. If your gel doesn't look like any of those, first you go back to your map. Make sure you didn't do anything crazy. You go back to the computer, make sure you got the right restriction enzymes. And then you might say, crap, it was 1,000 base pairs what it was supposed to be, but maybe it's not actually what I wanted. And then at that point we go, we're going to punt and sequence. What the hell's in there? Right? And then we just send it off for sequencing, and hopefully you get the sequence and it's right, and then you go back and go, oop, I drew my map wrong. I put the wrong, thing or something like that. Or we get the sequence back and we go, oh, this isn't what I was trying to PCR. We need new primers, right? Before we go to that next step, we're clear that what we have in PGC blue is really what we want. If the pattern all looks good, we just move on without sequencing. Because, the odds of two different things randomly having the same pattern and an orientation, it's within the realm of possibility, but it's not going to happen. I say that now, and then we'll be screwed. Okay, so does that make sense? Yes? We're in this middle? Okay. Thank you. Okay. So PGC, right, so it's in the PGC blue. We can cut it out with XHO and Hindi 3. There's a matching XHO and Hindi 3 site in PGL3. So now we call this directional cloning because it can't go in the other way. Having issues. Oh, I touched the button, I think. So, right, the Hindi 3 overhangs go together. The XHO overhangs go together. The XHO side's not going to want to, the XHO of the insert is not going to go to the Hindi 3 of the vector and vice versa. So now anything that gets in here should be in the right direction. We'll check again later, but we don't have to worry about, whereas the first one, it could equally go in either direction. Right, because it was just the GCNs to the Cs in there, and it could go forward or reverse. Okay. The thing that we as students and everybody tend to forget is there are other restriction enzyme sites in the PGC blue vector that end up carrying over into our PG3 vector, which is why I'm super crazy about drawing maps. And if you come to my office to ask me about your plasmid and you don't have a map drawn, I start screaming and yelling and having a fit. Really loudly. Yeah. Because we can't do any troubleshooting unless we know exactly what's in there. And as many times as I've looked at all these stupid vectors, I still can't memorize all the multi-cloning sites. Right? So if you were going to play our little game that you played before with KPN1, right, if this was our +1 here, remember we said KPN1 was there, and KPN1 is here, right, if we're going the right way, and then we're just looking in the PGC, or PGL3, right, it's got other cloning in here too. You say, oh look, here's a KPN1 site, out here, oh I can cut KPN1 to look for orientation again. Before I send it to sequence, we'll just make sure. And we want to make sure what's in the colonies have an insert, because PGL3 does not have blue-white. So all your colonies are going to be white, whether they're closing on themselves or not. So there's a little more screening because you don't have that nice blue-white screen. So you're like, okay, I'll just cut with KPN, because then I'll see what's in there and make sure it's in the right orientation. And then and so you look and you say, okay, I should, if I'm correct, this piece here is going to be 1,300 base pairs. I counted it all up. And then you run your gel, and you have two bands. And they're 300 and 1,000. And you're like, oh my God, I'm totally screwed. This is the wrong promoter, blah, blah, blah. You do it again, you get the same result, and then you throw everything away and you go back and make new primers and start all over. And then Malone says, "Where's your map?" And you bring it in and you show me your map, but it doesn't, you forgot. You just had your insert in PGL3, but you forgot these guys, because guess what? Remember that KPN1 that was in your multi-cloning site from PGC Blue? You carried that with you. And so, oh, those gels were right. Here's 300 and here's 1,000. But because you forgot to, you either didn't make a map or you just made it without being anal and crazy, you screw yourself over, right? And you might not realize that until you're actually making your map the very next, you made new primers, you went through all this shit again, and then you're making the map, and then you go, ooh, I was right the first time. Okay, so maps are super important because it always bites you in the ass. Sometimes it bites me in the ass, which is bad too, but it's usually the actual, you don't want to be in that situation. And then that's your whole promoter, and then that's the first round that we'd want to test to see, does it drive transcription? Do we get any light from that?